Dwayne, you want to come up here and we can answer any questions? In the first study with the tetanus toxoid, did everybody get the tetanus toxoid and half of those got the CMV, or did it was?
The other way around. Everybody got CMV, half got tetanus.
Okay.
Yeah, so they were randomized. They all got the same vaccine. And then we only did the imaging study at one vaccine. So the patients were randomized to either receive, prior to the imaging of their PP65 loaded DCs, they got either a tetanus booster 24 hours before the RNA loaded CMV DC vaccines, or unpulsed dendritic cells, their own autologous unpulsed dendritic cells, 24 hours prior to the CMV vaccine, and then we imaged the migration of both. So
it could have been the tetanus that made them survive longer.
it's possible. It's possible. So we did, the initial study was really to look at, of course, the migration. It wasn't until the unblinded outcome data showed the correlation with the migration and the outcomes. So I didn't have time to show, we did an extensive amount of then mouse studies to try to understand mechanism. So all I could say is in murine studies, tetanus toxoid has no ability to enhance the efficacy of a vaccine unless the vaccine is targeting an antigen
something
in the tumor.
Okay.
So we did all those studies in mice where you give them tetanus booster, you do increase migration. The pathways are the same as what we saw in humans in terms of chemokines that are elevated. But if you give a dendritic cell that either targets an irrelevant antigen or the tumor isn't expressing the antigen that the DC vaccine has, then there's no enhancing of efficacy. If it's targeting the tumor, it's definitively, at least in animals, works through enhancing the vaccine. But
Interesting.
In patients, it's hard to know for sure.
Minma?
When you use the PP65 mRNA to stimulate these things, do you know how high or whether they express that protein and the level of protein?
How much protein expression when we transfect them? No, we don't measure the protein levels directly. We've looked at the capacity for the RNA transfected dendritic cells to stimulate T cell responses in vitro. So they're very potent at activating and expanding PP65 specific T cells, but we've not correlated that to the level of protein expression.
So that means you think that the RNA that driven the DC response?
Oh, it's great question. So no, you need the appropriate protein peptides to stimulate the T cell response, and that we've shown with a variety of controls at both the target and stimulation level. But your question of whether RNA transfection itself may be activating in some way to the dendritic cells, certainly possible, and single-stranded RNAs in and of themselves are not extraordinarily immunogenic, but you can get some TLR activation. Double-stranded RNAs or in fragments of RNA are much more immunostimulatory. They activate some of the innate TLR signaling pathways in cells. So with the panel of markers that we look at for phenotype doesn't show a dramatic effect of RNA transfection alone, but it doesn't rule out that there's some activating component just to actually electroporating the cells. So it's a good question.
Hayden?
So in my review of the literature, which is incomplete, but it seems that the CMV proteins and DNA we found, if with having used pretty sensitive detection techniques in GBM cells, that mRNA, the actively replicating virus, things like that, cannot be. And I was wondering if that's the case, if my understanding is correct, how you would characterize the sort of current thinking around how CMV infects GBM cells.
You want to start?
Yeah, I think that that's why I showed that slide from this review article, and it talked about other herpes virus-related cancers, and they have this so-called abortive infection. I think in some B-cell lymphomas that are Epstein-Barr virus positive, they have no viral production. They have evidence of virus expression. I think the thought is that the virus is in this state that is not typically what you see in a normal kid who's infected with EBV or somebody who has a latent EBV. All of a sudden, the biology has produced this weird environment, and things happen with the virus that allow it to express gene products but not replicate and have virus. So I think in most EBV-related tumors, if you grow the tumor cells, you can't actually get live virus out of them either.
Yeah.
So it's kind of the paradigm is actually that there's not a productive infection in these virus-associated cancers, I believe.
Yeah. And I would probably add to that to say that, one, I would echo Charles' comments that if you look at the majority of clearly viral-associated tumors, even where there's a known etiologic link between the infection, the proteins, and how this driving oncogenesis, in the overwhelming majority of cases, you can't culture out live virus, and it's only a proportion of those tumor cells that harbor virus, even after oncogenesis in many cases. I'm not 100% convinced. I've talked to some virologists, I'm not a virologist, who've talked about clinical isolates, even from infected patients, are not always the easiest things to grow out and culture out. And so a very, very low subclinical level of virus may just be difficult to propagate. I don't know that we have definitive evidence whether there's no viral replication at all. But as to your question of whether RNA is detected, so we and others have, in those published papers, looked at detection of CMV RNA. So you can detect RNAs. You can detect RNAs across the spectrum of the viral life cycle as well. But I think we and others have failed to be able to culture out infectious virus or propagate virus in a way from supernatants or lysates and super infecting other cells.
And it's also been a big problem of people that send a tumor for RNA-seq and they say there's no virus gene expression. I don't know the answer to that because you can amplify it with PCR, but maybe it's just such a low level. And when you're looking for, it may be that it's only expressed in a subset of cells like the stem cells of the tumor that are less than 1% of the total tumor, so you're looking for a needle in a haystack. But it's been a confounding problem, and I think one of my grants recently got rejected because they'd seen this paper, and the paper said there's no virus there by RNA-seq, so how are we going to fund this research? Ralph.
So when you said early on in your presentation that 50 to 90% of people are infected with CMV, which tissues are infected?
So the characterization of the percent of the population that's infected with CMV is really based on serology. So that's you may have its own limitations, but it's the 50 to 90% epidemiologic data is based on the percentage of population that has circulating antibodies specific for human cytomegalovirus. There has been a lot of studies on, in a typical individual, you're not going to find much, if any, active replicating virus. But when people have looked for where the latent virus is harbored, it tends to be in progenitor populations within the bone marrow. There's not definitive data, but the suggestion is perhaps stem cells. There's even some good papers on viral genomes being able to be passed on to the progeny of stem cells without active viral replication. It's a good paper in PNS from a group at Stanford that's looked at that. So the general, and as Charles mentioned, in the brain, when people have looked, these have been more in animal models, but again, the progenitor populations tend to be where the virus lays latent, or at least are the cells that you can see early reactivation out of when you culture those cells under conditions where there's no immune suppression. But in a normal, healthy individual, you would be hard-pressed to find positive protein or RNA positive cells because it's present in a very, very trace population.
Is that what you said, that when they did the autopsies, that they were clear a Western blot analysis?
That's great. You would not, so in normal tissues, you would not expect to find much protein or RNA expression. I think the precursor frequencies, for instance, in peripheral blood mononuclear cells is known. It's 1 in 10,000 or somewhere in that range based on DNA amplification studies. But I mean, we're talking very, very trace in normal peripheral tissues, and you don't see much reactivation or gene expression outside of immunosuppression. But there's a lot of caveats that aren't known. So, for instance, since this virus is common in the population, it's obviously transmitted predominantly from healthy individual to healthy individual. So periodic reactivation and shedding of infectious virus happens all the time. That's how you get a relatively ubiquitous patient population that's infected. But it's the epidemiology is mostly based on serology, not detailed understanding of exactly where these cells are.
Some interesting information that leads to more understanding of where it is harbored. I mean, they look at the bone marrow stem cells, and that's typically where they'll find latent virus, but that's because they're easily accessible. But if you take a 2-year-old infant that gets infected at daycare, which is the most typical way in America almost now, then that child may secrete virus in their urine and their saliva and their feces, and it's in the blood probably, but primarily it's in anything that's secreted, and it can shed for months and months and months. Interestingly, the number one mode of transmission worldwide is breast milk. 100% of seropositive women secrete live virus in their breast milk when they breastfeed. If these are seropositive women who are non-immunocompromised, it's just the number one mode of transmission. So it must live in the breast ductal epithelium. So I think it goes everywhere, and kidneys, brain, salivary glands are primary spots.
So Wayne, also in your discussion of the 2 trials, PP65, do you have any idea, maybe possibly from biopsies, but how prevalent PP65 was in the brains of those individuals who were being treated? Like how many cells, like what fraction of cells have
PP65? It's varied. So we in those patients, that wasn't part of the eligibility criteria for enrollment, but we have looked by immunohistochemistry across a series of GBM samples, and you will find some tumor cells that show much more widespread detection in tumor cells, and others that are just isolated positive cells and surrounded by areas of negative cells. We were not able to see, at least in the patients that were enrolled in the study, and it's a small study, so with a small number of patients, trying to correlate level of expression with outcomes is difficult. So we have not been able to make an association between the levels of CMV detected in the tumors and response to therapy. What we have seen as a correlation is either the migration of the cells to lymph nodes has been a much stronger correlate than anything else we've looked at. And in the most recent study, the magnitude of immunity of PP65 specific immunity fold change over baseline. So in other words, what the magnitude of immunologic response was to the vaccines has been a much better correlate with outcome. But the amount of detection in tissues at baseline has not been, in the limited studies we've done, dropped out as a strong correlate.
So would your vaccine be just as effective if you use a different epitope, not even the CMV, if some protein is prevalent on cancer cells?
On cancer cells. It's a good question. You could argue, so I don't know, we haven't tested. There's a couple of potential arguments that either for or against that. So one would be that, self-antigens, or that we've had a lot of vaccine studies that have targeted overexpressed tumor-associated antigens. And one potential limitation may be that those may be antigens that you also have a higher degree of tolerance against because they are self-antigens. And so stimulating when studies have looked at the T cells that respond against self-antigens, if we call them our TAAs, they tend to be lower affinity, lower avidity T cells, presumably because of some deletion in central tolerance. So there's at least a theoretical argument that neoantigens, either true mutations that are new or foreign antigens, viral, et cetera, maybe have a much better prevalence of high affinity T cells that can react against it. So you might argue that we don't think we would see the same both magnitude of immunity or potential affinity or avidity of T cell response, but we have no comparative data to say that. That's just at least an argument that we thought made this an attractive target to explore. But it's a great question. I think it's so we are very interested in actually doing a combined study where we would actually ask that question directly, vaccinate against a tumor-associated antigen that's overexpressed and CMV antigen in parallel, look at ability to generate immunity using the same platform, and also relationship to outcome. But I don't have any information, but it's a good question.
So along a slightly different language, back to this idea of pregnant women with CMV. If 100% of those women, CMV positive women, secrete CMV through their breast milk, and a majority of the population is CMV infected, is there some sort of immune priming through that breast milk as well, specific to CMV, that's preventing these immune naive, infants from developing complications to that?
Well, I think you've just tapped into a massive area, and we have a colleague who was at UCSF working with the Wiemels lab, Joe Wiemels, who is. Were you here when he came and talked? Steve Francis. He believes that CMV infection in utero is now linked to leukemia. They had a published paper about 3 months ago that got quite a bit of press, and they showed that for children infected in utero, they did a agnostic screen of bone marrow from all these kids with leukemia, the bone marrows, looking for every infectious agent known, and then CMV popped up as a very high relative risk factor. And then they went back to the heel blood sticks from those kids when they were born and found that they were all infected at birth. So they had probably been, they had been infected in utero. And he thinks that the infection in utero may screw up the normal immune response, because that's when you're actually developing your immune recognition of self and non-self, and that timing may reprogram the immune system. Interestingly, these children that get leukemia are prone to multiple infections throughout their lifetime before leukemia happens, suggesting they have altered immune functions. Anyway, but so I think that, I don't know if that's the right answer to your question, but I think there's probably something very critical, just like learning a language. If you're exposed to something at the right time or the wrong time, you may have a totally different immune response. I think in general, most kids exposed to CMV, or people exposed during early childhood or whenever, have no problem with it. But obviously, if you get exposed when you're immunosuppressed, or if your immune system's developing, it may have a totally different effect.
Yeah, there's also, I think you have to take into account that most of our description of the importance of CMV infection is driven a lot by the clinical observation, which is we don't see severe disease when you're acutely infected as a normal person, and we don't see severe disease except in very strongly immunosuppressed patients. So we say it's a relatively benign infection unless you have these complications that result in severe immunocompromise. If you look, though, at the biology of CMV infection and how it affects the immune system, an immunologist would not say that this is a benign infection. So a huge portion of your immune system is devoted to suppressing CMV. In a normal individual, you can see upwards of 5, 10, 15% of their circulating T cells sometimes being devoted to trying to, they're at least CMV specific, implying there's this chronic expansion and chronic stimulation of these T cells. There's also a phenomenon known as CMV-induced immune senescence. So as you follow older people who are CMV seropositive compared to CMV seronegative patients, you can certainly see that the T cell compartment in terms of exhaustion and some of these other phenotypes can be driven quite significantly by CMV infection. And so those people may never come down with a symptomatic, clinically significant CMV infection that we classically see, but it doesn't mean that the presence and the chronic stimulation of the virus is not having profound systemic effects. They just don't manifest as an acute infection in most cases. So I think we don't understand all of the significance of harboring a lifelong immunogenic chronic infection, but it certainly it skews the immunologic compartment quite significantly.
Yeah, that's why I've wondered if this chronic, maybe people that have glioblastoma associated with CMV have had some altered immune function prior to this, either due to CMV or something else, because we know in a paper the Reardon paper, that when they looked at the CD4 counts of GBM patients, they were average below 200, which is the definition of AIDS for HIV. So has that panned out? And
Yeah, the lymphopenia. Yeah, so we entered in this with the hypothesis that it was interesting. If you look at the literature in CMV virology, a lot of one of the most permissive cell lines, one of the commonly permissive cell lines used to grow the virus, even in a laboratory setting, are glioblastoma cells. So not every single cell is very good at propagating CMV even in laboratory setting, but gliomas seem to be particularly good at it. And so we favored the hypothesis that this immunoprivileged microenvironment is just a good place for a virus to kind of hang out and avoid the immune system. And I've sort of become, as I've learned more and more about what else the virus does, become more intrigued by the immunomodulatory and potentially oncomodulatory properties of the virus. But there's other factors in patients with glioma, like lymphopenia, that has been described and published. Peter Fecci at Duke, as a grad student, published a paper that looked at CD4 T-cell lymphopenia in newly diagnosed GBM patients. Others have shown there's profound cell-mediated immune deficits in these patients. And we all know that cell-mediated immunity is one of the major components for suppressing CMV reactivation. So it does become a question of what could come first. Could chronic infection be driving immune senescence and playing a role in also glioma biology, or can glioma biology be causing immunosuppression and providing an environment for reactivation? I think both of those are possibilities, and I don't profess to know which may be more likely, but I think there's a lot of factors that could contribute to low-level viremia in patients or low-level viral reactivation. Good question.
So we keep talking about glioma, but I think what to me was most surprising about the encephalitis paper that was published is that they found more patients who had symptomatic CMV encephalitis who had brain mets. So these were not people with gliomas.
Right.
Most of those people, from what was described in the paper, were not on systemic immune therapies, were not immune suppressed in the way that you would normally associate with CMV. So I think that was even more surprising, and I'm curious what both of you thought about that.
I think that, I've been thinking about that paper. I don't know. I mean, if you have a cell that's got latent CMV and you want to reactivate the virus, shoot it with radiation.
Yeah.
So we do know that Mets are positive as well. But I think that just the fact that you have radiated the body, and stressed the system, just like if you give someone radiation, they might get cold sores or reactivate zoster. It may be just this cytokine storm that, all the herpes viruses just shoot up and become. But that paper kind of really shocked me that about 25% of patients have, according to that paper, get CMV encephalitis, which can be fatal if not detected and treated. And it, as the neuro-oncologists like Mike and Santosh will know, sometimes you see these patients who, and that don't seem to have dramatic tumor growth, or maybe I'm just making this up from my memory anecdotally, but patients don't seem to have progressive tumor, but they get sick and just kind of dwindle. And I wonder if that's what some of these people have. And so maybe that's why some of these patients do better on Valcyte. I don't know if they do.
Yeah, I think the surprising finding of the paper wasn't that. So as Charles mentioned, radiation, basically if you think of anything that induces significant cellular stress, can cause reactivation of latent CMV and other herpes viruses, and that's been known for some time. So I think the surprising finding, if you will, from the paper was that this occurred, and we don't really ever think about this spectrum in patients routinely. It's not something it's on the radar for transplant patients all the time. But again, that's because they progress if you don't treat to very significant morbidity and infectious complications, which I think most people would argue have not been seen in patients with glioblastoma. And I don't think likely just missed, although maybe, but it's not something that's routinely thought about or looked for as a risk factor in patients. So I think it was more the finding that they found not just that you could see reactivation by irradiation, but that seemed to be coincident with neurologic symptoms in patients, and that treatment seemed to have a benefit. Now, this is not a randomized prospective controlled trial with all those caveats, but just the fact that there may be clinically significant, I would call it, subclinical or not diagnosed, but still could be significant for patients and that treatment intervention may have some benefit to those patients wasn't previously appreciated. And I think that's the more surprising and interesting finding, which hopefully will be followed up in a much larger, prospectively designed study to understand that.
I mean, it would be interesting. I know we've started looking at plasma PCR for CMV, and you've had at least one patient who had circulating CMV prior to surgery. It would be interesting to follow some of these people and see if they actually have reactivation during radiation and Temodar. Because as I said, any normal person that's in the hospital, if they have viremia of CMV, is highly unusual, and that can be associated with, a high increased risk of other problems.
So there's your two trials. So concerned about these cells that don't express p65 or the antigen. So could they be likely to escape the therapy? Is that possible? Or do you try to induce lysis in the entire tumor, or induce a living stage to the virus so that it spreads within the tumor?
Spreads within.
And eventually all the cells are expressing p65.
Yeah, it's a great question. So the two things that we know that all of the tumor cells don't express, at least by the detection methodologies that we have at the start in all patients. And in the patients that did relapse that we did have biopsies on, we did not see antigen clearance. So you could still detect antigen present in recurrent tumors, not eradicated with vaccination. I talked to a lot of people in the transplant field, and they said, they don't think you can ever eliminate the reservoir of CMV in the body. And so, you could either argue that those tumor cells were resistant through some other mechanism, or can re-express or become reinfected. You've got a systemic infection that at least for my colleagues, they say, I don't think you're ever going to clear it. So I think to improve targeting, we need to do either combine with other antigens. We're very interested in first confirming the clinical activity of the treatment. Your question really gets at mechanism and how, if it is working, then how is it truly working in patients? And although we have our hypothesis, I think we have to be open to the idea that these vaccines induce a number of things: cytokines, NK cell activation, antigen-specific T cell responses, CD4 and CD8. So some that can directly recognize tumors, some that actually act as helper T cells to other immunologic responses. And in truth, it would be the best we'll be able to do is correlate different components of that immune response with outcomes to try to say this is what we believe is the most important mechanism, because it seems to be the strongest biomarker for outcome. But we also have to be open to the idea that I would likely not know in patients if any one of those modalities, NK cell activation, cytokine, or direct killing T cells or bystander helper cells, could all lead to a better anti-tumor response. We're trying in animal models to dissect that out, but in the trials it's a little bit harder to know. And so
If you added a radiation step, like Charles was just saying, everything is, who knows what happens, but it might induce lysis throughout the tumor. Then you have spreading of the CMV to every cell. I don't know if that would help or not. But.
Yeah, you're right. I think we've thought more about, and probably because that's the way the field is going, some of these combinations with other things like checkpoints and other things that sort of get at the immunosuppressive axis in a more way, as well as expanding the number of antigenic targets. But you're absolutely correct. There's a lot of other components that we haven't addressed and we could, and so it's a good suggestion. We got our hands full with what we're trying to get this phase 2 trial done, but I think those are.
Yeah, it's a problem is you get a concept and you plan a trial on year 0, and then you finally get it into the trial 3 or 4 years later, and then somebody says, well, did you think of all these new things we just learned in the last year? And you're like, well, if you could just go like that and start another trial, but unfortunately it doesn't work like that. Just getting the money is
yeah.
It takes 5 years.
Yeah. No, we think if we can hopefully confirm in a prospective randomized trial that there's clinical activity, that one, there's a lot of biomarkers you can look for to ask what components do you think are most important, and then the combination, opening the door to combination strategies, becomes a lot more attractive when you feel like you've got a baseline of something to work from. So I think we've thought about all of those things. We just haven't incorporated any of them yet into a clinical design.
So do you have a good animal model that's translatable into the clinic, or are they not predictable? Are they not predictable as human?
So one of the limitations, at least in modeling CMV infection in an animal model, is that the capacity to generate, you can certainly overexpress a protein or two. We've screened for MCMV infectable lines that then maintain and harbor a persistent infection. But none of them directly model quite well that condition. So they're contrived, and no matter how you do them, if you implant already actively infected CMV gliomas into animals, they mount a pretty strong immune response that ends up rejecting. So you have to go through some manipulations to allow the cells to grow. Nino Chioka and his lab group, I think, has worked on an elegant genetically engineered mouse model where they can show that previous infection of those animals with MCMV can, the tumor genesis can be accelerated in the MCMV infected animals, and you do see at least a reactivation of MCMV in those models. But every one of them have their limitations. So we, yes, we have those, but I don't know how forward informative they are.
What I've found is that no matter how many things you do in a dish or in a mouse, people are still going to say, so, and to a certain extent say, yeah, so how does that apply to humans?
Yeah. We try to use them to prioritize strategy. So if something looks more promising in the murine model, we will use that to prioritize, hey, if you had two choices of what to take into the clinic. But I think I've always said that we do phase 2 trials that sometimes don't inform us how the phase 3 trial is going to work out, even in humans. And so humans aren't even great models sometimes of outcomes in humans, and at least in smaller trials to larger trials. Otherwise, we would never have any that fail at phase 3. So we've tried to use the murine models as prioritization and mechanism, but less for prediction, because I don't have a lot of faith.
Is the
in it.
phase 2 at Duke and
It's open at UF now, and it'll be open at Duke as well shortly.
so that you'll get 120 people, hopefully not in too long a period of time?
Yeah, hopefully not too long.
Yeah.
Yeah.
Well, I think that it's 4:40 and it's time to stop and let people go that are leaving this portion, and then the rest of the folks are going to hang around for the evening session. You can have time off and have dinner, and then we'll be back, I believe, what did we say, at 6 p.m.? 5:30 for registration, 6 p.m. for the panel discussion. And once again, thank you all for the people who have come from around town or out of town, and especially Dwayne, who came from the other side of the country. Thank you so much. He's got 3 little kids he's got to get back to.